You have 3.2 billion letters of code inside you, a unique blueprint that makes you, you. This is your genome, and it's being read right now by your cells, which are using it to create proteins. Proteins do everything: they build, repair, and even kill. They're the workhorses of your body. For instance, the protein hemoglobin carries oxygen in your blood, and a defect in its code can lead to sickle cell anemia, affecting over 300,000 births worldwide every year. You are your genome, and it's fascinating to think about. But what if you could edit it? Change the code, fix the mistakes. It's a tantalizing prospect. Did you know that there's a type of jellyfish that's immortal, and scientists think it's because of its genome? The Turritopsis dohrnii, also known as the immortal jellyfish, can transform its body into a younger state through a process called transdifferentiation. This has sparked interest in understanding how its genome contributes to this unique ability. As you consider the possibilities of genome editing, you're not just thinking about fixing diseases, you're thinking about rewriting the fundamental code of life itself. Meet CRISPR, a game-changer in genetics. It's like a word processor for DNA, cutting and pasting with precision, guided by RNA. You can find the right spot and make the edit with ease. For instance, scientists used CRISPR to treat sickle cell anemia in a patient named Victoria Gray, who received a revolutionary gene therapy that edited out the faulty gene responsible for her condition. The results were astonishing: her symptoms disappeared. But how does it work? You use a guide RNA to find the target, like a GPS navigating to a specific address. Then the enzyme cuts, and the cell repairs itself. You provide the template, and the cell does the rest. Interestingly, CRISPR is not just limited to humans, it's also being used to develop more resilient crops, like corn that can withstand droughts. Surprisingly, the CRISPR system is based on a bacterial defense mechanism that's been around for millions of years, and it was only discovered in the 1980s. As you delve deeper into CRISPR, you'll realize its potential to revolutionize various fields, from medicine to agriculture, and it's an exciting time to explore its possibilities. You have trillions of cells, and each one has a genome that's made up of DNA, which is composed of nucleotides. These nucleotides are like letters: adenine, guanine, cytosine, thymine. CRISPR uses them to edit your DNA with precision. It's like a molecular scalpel, making precise cuts guided by the enzyme Cas9. This enzyme is the key to making CRISPR work, and surprisingly, it was discovered in a bacterium that lives in the hot springs of Yellowstone National Park. For instance, scientists at the Broad Institute used CRISPR to edit genes in human cells, with a success rate of over 90% in some cases. Your body is making Cas9 right now, and it's being used in labs around the world to treat diseases like sickle cell anemia. You might be amazed to know that the human nose can detect over 1 trillion different scents, and scientists are now using CRISPR to study the genes that make this possible. It's a powerful tool that's opening up new possibilities for medical research and treatment. With CRISPR, scientists can now edit genes with unprecedented precision, and this is just the beginning of a new era in genetics. CRISPR can edit human embryos, a revolutionary concept that's changing the landscape of genetic engineering. You can fix genetic diseases before birth, which is a significant breakthrough. For instance, a study published in 2017 used CRISPR to edit out a heart condition in human embryos, with 42 out of 54 embryos showing the desired edit. That's a staggering success rate. But it's not without risks, such as off-target effects, where unintended parts of the genome are altered. Mosaicism is another concern, where some cells in the embryo are edited, while others are not. Germline editing, in particular, raises eyebrows, as it involves making changes that can be passed down to future generations. Surprisingly, did you know that CRISPR can also be used to bring back extinct species, like the woolly mammoth? The possibilities are vast and complex. As you consider the implications, you have to weigh the benefits against the potential consequences. You must think about the long-term effects on human health and the ethics of playing with the fundamental building blocks of life. The concerns are real, and it's crucial to approach this technology with caution and careful consideration. You might be using CRISPR-edited products right now. Food. Medicine. They're already here. Gene-edited crops like corn and soybeans are being grown in the US, with over 90% of these crops being genetically modified. For instance, a company like Monsanto has developed CRISPR-edited corn that's resistant to the corn rootworm, reducing pesticide use by up to 50%. Better yield, less waste, it's a game changer. You're eating them, and they're in your body. Changing your genome, not directly, but indirectly, the effects are real. You see, when you consume these gene-edited crops, the modified genes aren't directly altering your DNA, but they can influence your gut microbiome. This can have a ripple effect on your overall health. Surprisingly, did you know that gene editing can also be used to bring back extinct species? Scientists are exploring the possibility of using CRISPR to revive the woolly mammoth. It's mind-boggling to think about. As you go about your day, you're interacting with CRISPR-edited products in ways you never thought possible. The technology is advancing rapidly, and it's crucial to stay informed about its implications. You're part of this genetic revolution, whether you realize it or not. Most people think CRISPR is a cure-all. It's not. It's a tool. A powerful one. But limited. You need to understand its boundaries. For instance, scientists used CRISPR to treat sickle cell anemia in a patient named Victoria Gray, who received a groundbreaking therapy that edited out the defective gene responsible for her condition. This is a prime example of CRISPR's potential, but also its limitations. It can only target specific genetic mutations. It can't fix everything. Only genetic diseases. Not all of them. And it's not without risks, like unintended edits to the genome. You have to consider them. Surprisingly, did you know that CRISPR can also be used to bring back extinct species, like the woolly mammoth, by editing the genes of its closest relative, the Asian elephant? What are the alternatives? Other therapies, like gene therapy, which doesn't involve editing the genome, or other treatments, like medication and lifestyle changes. These options may not be as flashy as CRISPR, but they can be just as effective. You should weigh the pros and cons of each approach. It's a complex decision, and one that requires careful consideration of the potential benefits and risks. CRISPR can also edit human brains, a surprising concept that opens up new possibilities. You can change behavior, fix neurological diseases like Parkinson's, which affects over 10 million people worldwide. For instance, scientists have used CRISPR to treat a patient with sickle cell anemia, a genetic disorder, by editing their stem cells. But it's not without risks, and unintended consequences can occur. You have to consider them, weighing the benefits against the potential downsides. What are the implications for medicine, for society? You have to think about it. Interestingly, did you know that our brains produce new neurons throughout our lives, a process called neurogenesis? This fact challenges the long-held idea that our brain cells are fixed. As you ponder the possibilities of CRISPR, consider the case of Jennifer Doudna, a pioneer in CRISPR research, who has spoken about the need for careful regulation and ethical guidelines. The potential is vast, but so are the unknowns. You must think critically about the consequences of editing human brains and how it could shape our future. The more you learn, the more complex it becomes, and the more you realize that this is just the beginning of a much larger conversation. CRISPR is a powerful tool, capable of editing your genome with unprecedented precision. It can fix diseases, like sickle cell anemia, by correcting the faulty genes that cause them. For instance, a study published in the New England Journal of Medicine used CRISPR to treat 10 patients with sickle cell disease, resulting in a significant reduction in symptoms. But it's not without risks. You have to consider the potential for unintended consequences, like off-target effects. You might be surprised to learn that CRISPR can also be used to bring back extinct species, like the woolly mammoth. What's next? Will you use CRISPR to edit your own genome or that of your children? The future is uncertain, but one thing is clear: CRISPR is here to stay. It will change your life and the lives of those around you. You'll have to navigate the complex ethics of gene editing, weighing the benefits against the risks. As you consider the possibilities, remember that CRISPR is not just a tool, but a key to unlocking the secrets of life itself. The question is, how will you use it?